Okay, I think we'll get started. Well, good morning and thank you for joining our webinar today. Ordinarily, I would like to have been able to meet face-to-face with investors and answer questions given the significance of yesterday's announcement. But given our collective circumstances, we thought this webcast would be the best way for our shareholders to hear from me directly. Before getting started, I'd like to recognize that over the recent months, we've welcomed many new investors to the IBX register. And I'd like to extend our thanks to all IBX investors who continue to support our business and simply say, I look forward to meeting you in person sometime soon. As most of you are aware, the study has been an area of intense operational focus for Imagion and will be an important milestone for our business and we hope an inflection point for investors. This announcement signals another important step forward, reaffirming we remain on track to initiate the study this year. Over the next 10 minutes or so, I will try to provide some further insights regarding the announcement and particulars about the study itself, and then answer questions that have been submitted. You may submit questions throughout the presentation using the Q&A button on the bottom of your screen. We'll try to address as many of the questions as possible. So first, this study is what would be known in the US as an early feasibility study, or a phase 1 study in Australia. And like all phase 1 studies, a primary goal of this study is to determine whether our MagSense nanoparticles are safe for use in humans. Based on the toxicology study we completed late last year and the known history of use of iron oxide nanoparticles, we expect to meet this primary endpoint for safety. But the study is also intended to provide initial insights regarding the effectiveness of our HER2 targeting nanoparticles for determining the presence of and spread of HER2 breast cancer in lymph nodes. And to be clear, this is primarily a test of the MagSense nanoparticle formulation and will use our existing preclinical research machines for measuring of the nanoparticles. There's no point in spending money now on new machines until we know the nanoparticles work as intended. This first study in humans will help us to know how quickly we can move towards a more complete study of the overall system for commercial clearance. As most investors know, we have historically expected to undertake the study in the US under a device exemption from the FDA. This strategy had largely been driven by the fact that we had our MagSense instrument technology at both our lab here in San Diego and at the MD Anderson Cancer Center in Houston. And we believed we needed to have an instrument on site or at least in close proximity to where there would be a large patient population accessible for enrollment. Over the course of this past year, as we developed the clinical study plan further, we realized having our machines on site would not be a requirement. This is what has allowed us to revisit undertaking the study in Australia, where there's a very favorable regulatory and clinical environment for human clinical research. For the Australian study, we've established that we can send the samples back to our lab for analysis, saving the expense of having our instrumentation at the clinical sites. Our goal is to test our tumor targeting nanoparticles in approximately 15 to 20 patients, but we are planning to make enough material should we or our study partners wish to continue with additional test subjects. Given the smaller accessible population in Australia compared to the US, we're planning to use 3 to 5 sites in an effort to minimize the length of time to undertake the study. At present, our plans are aligning in such a way that we are hopeful that we can start enrolling subjects in the 4th quarter of this year. Importantly, we are expanding the scope of the study to include 2 detection modes: our proprietary magnetic relaxometry technology and magnetic resonance imaging, commonly referred to as MRI. The ability to include MRI in this study was one of the contributing factors for focusing on Australia. Quite simply, it would have been more difficult to add MRI to the study in the US and risk adding time and cost. And alternatively, we would have incurred added costs to do a completely separate MRI-only study. We believe we can achieve significant value by being able to assess both modes in one study and at a reduced overall cost. Our collaboration with Siemens Healthcare in Australia was a precursor to this decision. Although their contribution is technical in nature and not financial, their expertise to identify the optimal MRI scanning protocols and provide technical support to the clinical sites is crucial to adding the MRI component of this study. Once this collaboration was secured, We were comfortable with the change of plan, and we've moved quite quickly. There are a number of other additional advantages related to running the study in Australia. Investors may recall that late last year we formed our scientific advisory board, and we were able to attract some very highly regarded Australian researchers and clinicians. This has added weight to our organization with regard to the Australian clinical landscape. Undertaking the study in Australia will provide the opportunity to strengthen our relationship with these advisors and the Australian medical research community at large. And of course, there are a number of strong financial benefits to doing the study in Australia as well. Firstly, we eliminate any currency exchange rate losses that we would have incurred doing the work in the US. And secondly, it simplifies the R&D tax incentive filings, eliminating the overseas filing component for these costs and making it likely we will benefit from the R&D tax credit. Furthermore, undertaking the study in Australia does not create any disadvantages on a go-forward basis from a regulatory perspective. We expect the data we generate in the study will be helpful as we proceed with further studies in other jurisdictions, such as in the US and the FDA. We also felt that undertaking the study in Australia could be a sound risk mitigation strategy, since so far Australia has not been as hard hit by the COVID-19 pandemic as the US. And although the pandemic has not set us back to date, we are mindful of the associated risks, and at least as of today, are seeing clinical research in Australia less impacted than at least in the United States. And though not a driving factor, we're cognizant that we are an ASX listed company with many Australian investors, and conducting the study in Australia creates an opportunity for IBX to contribute back to the Aussie community and the economy. So where are we? At present, we have principal investigators at 3 clinical research hospitals lined up, and we have designed the study to allow up to 5 sites in total. We've engaged a contract research organization to assist us with getting the study sites under contract and managing the study once initiated. Once we have contracts in place with study sites, we'll be able to announce those study sites. We will be submitting our application for the phase 1 study to a Human Research Ethics Committee for review in place of the investigational device exemption we needed for the US. We'll continue our dialogue with the FDA under our breakthrough device status for future US submissions, but we're not dependent on the exemption for the Australian study. Our ability to start the study is, of course, subject to the HREC approval. Manufacturing of the MagSense nanoparticles will continue as planned, and I can report that that stays on track. And based on our understanding of the HREC review process and our manufacturing plan, we could begin enrolling subjects for the study in Q4 of this year. So in conclusion, I'd like to remind you of some of the advantages of our targeted MagSense nanoparticle technology and why we selected HER2 breast cancer as a first indication. HER2 breast cancer is one of the more aggressive forms of breast cancer, but there are good treatments if the cancer is found early. An accurate, non-invasive, and non-radioactive imaging technique that could confirm the presence of cancer, not merely identify suspicious nodes, would be better for patients because it fits into the current clinical workflow, and it removes the need for unnecessary biopsy or surgical intervention, and thus eliminating costs and unfortunate side effects such as lymphedema. Demonstrating that our targeted MagSense nanoparticles works in HER2 breast cancer is just the first proof of principle and will allow us to invest in other cancer targets with confidence. With this study showing how our particles work for both relaxometry and MRI, we could have multiple ways to commercialize our nanoparticles for image-based diagnostics.